But for the moment, let's move to Jeff Calhoun. He's our next speaker today, and he's an associate professor at the Northwestern University. And his research interests focus on improving the genetic diagnosis of epilepsy through genome sequencing, machine learning, and functional characterization of variants of uncertain significance. And the title of his talk today is mTORC1 functional assay reveals SZT2 loss of function variants and a founder inference deletion. So Jeff, whenever you are ready, we can start. I think you are muted, Jeff, sorry. No.
How's that?
Now, yeah, right.
Perfect.
Thank you.
Sorry about that. Well, thank you so much for the kind introduction, and to all of the VEST organizers for the opportunity to present today. So yeah, let's jump right in. So I think sort of classically in our physiology courses, we're sort of taught to think of epilepsy as due to an acute insult. I guess maybe it's really easy to imagine somebody has a car accident, they hit their head on the steering wheel, you get an acute insult to the brain, a traumatic brain injury that later causes the development of an epilepsy due to that acute insult. But one thing that we've sort of realized as a field since sort of the mid-90s is that many cases of epilepsy actually have a genetic etiology rather than sort of the classical acute insult paradigm. And because of that, there's been a lot more interest in pursuing the genetics of epilepsy, and those pursuits have been quite fruitful. They started, as I mentioned, the mid-90s, and progress was a bit slow just because lack of next generation sequencing meant that in order to identify a new epilepsy-associated gene, you had to find a large enough pedigrees so that you could map variants between individuals with shared phenotypes. That being said, many researchers were very successful in identifying genes that are incredibly important to the field to this day. But one thing I think that is really easy to appreciate is that right around when next generation sequencing, or NGS, was launched in sort of the early 2000s, early 2010s, the identification of epilepsy-associated genes sort of skyrocketed, and this is true for other genetic disorders as well. And it's partially because we collected larger cohorts, and partially because just the technology meant that we could screen many more individuals at a much more rapid pace and more cost effectively. And I think what this means is that it's complicated. Now we know that hundreds of genes can cause epilepsy, so part of it can be a little overwhelming, but you can think that in terms of precision medicine, this is super useful because we can try to find shared pathways between multiple genes of the ones that are listed on this slide and try to develop broad strategies to tackle novel treatment strategies. But you can also think about at the level of each of these individual genes, could you develop a precision medicine approach that's very tailored to the perturbation of that one gene? So that's sort of a little bit more forward thinking. Sort of the immediate impact of this gene identification is in the clinic right now. Way more individuals get genetic sequencing or genetic testing, and so this is both good and bad outcomes. The good outcome is we have increased diagnostic yield, and that's fantastic. We can give an answer to individuals. They can potentially get on a precision medicine sort of treatment plan that's different from the treatment plan that they would have had, at least for some genes. We're still working on that for many of these genes. The downside is that we are much more likely to have a result of a variant of uncertain significance or a VUS. And so sort of like looking at the field right now, it seems like there's sort of these two, well, more than two, but at least these two major challenges. We need to continue to develop personalized medicine approaches to treatment, but also we need to figure out how do we solve these VUSs, because you need to know which individuals are candidates for a precision medicine therapy. And so that's sort of my focus at the moment. And I think it sort of aligns with the focus of the AVE group in general. And so now that we know that epilepsy is often of genetic etiology, what kind of genes are we talking about? I think it's not hard to imagine that many of the genes are ion channels. These are genes that directly impact neuronal communication. And so you can imagine mutations in these genes can perturb networks and cause hypersynchronous brain activity. But what I wanted to highlight here is that there's actually a number of biological pathways that are impacted that are not ion channels. And there's already many labs working on resolving VUS as ion channels. And so I think sort of the niche that I'm hoping to develop is working on sort of these non-classical, non-ion channel genes. So the group of genes that I'm going to talk about today are a subset of the intracellular signaling group, including these four genes and others, which I'll show in a little bit. These are all part of the mammalian target of rapamycin signaling pathway, or the mTOR pathway. And because they're all in the same pathway and they all cause genetic disorders, they're sort of lumped into this group of mTORopathies. So I won't give a lot of detail on the mTOR pathway because it's rather complicated, and you could easily fill up 25 minutes on this slide alone. But I did want to briefly introduce sort of a few major concepts. One of those being, at least from a genetics perspective, what I find interesting about this pathway is that genetic variants in different genes within the pathway cause distinct genetic disorders. So if you have genetic variants in the tuberous sclerosis complex, it gives rise to tuberous sclerosis. Whereas if you have variants over here in the GATOR1 complex, you get focal epilepsy. Sorry, I just realized I don't have the laser pointer. Is that, or at least
we can see your cursor.
Okay, perfect. Yeah. So the variants in the GATOR1 complex lead to a very different phenotype of focal epilepsy. And so I think that's interesting. One I also wanted to highlight is this PMSE phenotype. The actual phenotype isn't critical for today's talk, but it's caused by autosomal recessive variants in this gene strata. And it just so happens that it was identified in an old order Mennonite population as a founder variant. And so you can go from sort of autosomal dominant, relatively common tuberous sclerosis to very rare but elevated frequency in certain populations, variants like strata and PMSE. For the purpose of today's talk, we'll focus mostly on this gene SZT2, or seizure threshold 2. It's a very hard gene name to say. I apologize. You'll have to hear me say it a few times. It is a member of this complex called the KIK-STOR complex, and it functions as a scaffolding molecule. I'll get into the specific function in a little bit more detail in a few slides, but I did want to mention that it localizes to the lysosome and it's involved in the regulation of mTOR activity. The last thing I want to mention on this slide is there's some very common readouts of mTOR activity, and I wanted to highlight two of them that are going to be used in my functional assays. So one of them is the direct phosphorylation of S6K1, or S6 kinase, by mTOR. And so you can measure activity by the amount of phosphorylation of this protein. And then it also phosphorylates a downstream target called S6. And so measurements of phosphorylation of that protein is also a measure of mTOR activity. And depending on the exact assay you approach, sometimes you'll use S6K1, sometimes you'll use S6. Okay, so the questions that we wanted to address in this study were, first, can we design and validate a functional assay to identify pathogenic variants in this gene SZT2? And then if we're able to do that, does this actually have any clinical applications, such as allowing us to resolve VUSs in SZT2 and potentially impact patient care? So a little bit more on SZT2 associated epilepsy. Just like strata, it is an autosomal recessive condition rather than an autosomal dominant, which is the case for some of the other mTORopathies. There's been quite a few case reports of individuals at this point, the first one being in 2013, and then sort of a few a year every year since then. And most of the variants that have been reported in the literature are clear loss of function variants due to premature truncation of the SZT2 protein. And the sort of core features of these individuals include an epileptic encephalopathy with early age of onset, focal seizures that are often refractory to anti-epileptic medication, developmental delays, MRI abnormalities of the brain, and macrocephaly. So one of the things that we sought out to do in this study was do an expanded cohort, because we felt like it was impossible that all of the variants in this gene that were causing disease would all be loss of function, like clear premature truncation variants. We felt like there had to be missense variants that were out there that just weren't being published. And for good reason. It's really hard to know what to do with a missense variant in this gene for a couple of reasons. One, that it's autosomal recessive, so just looking at minor allele frequency in NOMAD isn't really that helpful. Two, it's a gigantic gene, so a single missense variant somewhere in the middle of the gene, what the heck could that do? And it also, this gene happens to lack any known functional domains. And so you can't even say that, oh, you have a variant at this particular position. It's more likely to be damaging because that part of the protein does a certain function. Well, we have no idea. We know overall what the function of this protein is. We don't know anything about the subdomains. And so we put out the word both locally here at Northwestern to both our adult epilepsy team and our pediatric epilepsy team, as well as internationally through GeneMatcher for any unpublished individuals that had either epilepsy or a neurodevelopmental disorder without epilepsy that had genetic testing and SZT2 variants reported. And so what we ended up with was a cohort of 12 individuals. With the exception of one individual, all of these individuals had at least one missense VUS, or in the case of this Valley 1984 deletion, an in-frame deletion. And so this cohort to us sort of represented a unique opportunity in that we could develop a functional assay to resolve these VUSs relative to the previously published cohorts, which are all pretty clear loss of function variants. The other thing I want to mention before I move on from this slide is just that there's quite a few variants that are present in the general population in NOMAD that you could use as a benign control for the development of a functional assay. We selected this one, proline 446 serine, because there's many individuals that are homozygous for this particular missense variant. It's highly likely to be a benign variant. Lastly, not our study, but a little bit of added motivation for doing this work. One of the major genetic testing companies in epilepsy is this company Invitae that started this behind the seizure program. It's a very popular program. They'll do genetic testing for pediatric epilepsy at no charge. And so it's been widely used, and we were very happy to see that they published their data and made it available so that we could see what the results were. And we were pretty astounded by this particular result. So they tested SZT2 on their gene panel in over 9,000 individuals, and they reported a result out 6% of the time, so over 550 results. And of those, almost all of them were VUSs. And so these are all clinicians, genetic counselors, and patients that have been reported, okay, you have this variant that we don't really understand in SZT2. Do you want to pursue this at all? Do you ignore it? What do you do with it? And so this was just sort of added motivation for us to try to figure out how we can resolve these VUSs. So I want to give a little bit of a cellular model for how SZT2 does regulate mTOR activity. And this is based on a back-to-back publications in Nature in 2017, which was our first indication of what SZT2 did. So it turns out that it's specifically involved in amino acid sensing, which is a particular arm of the mTOR pathway. And so a really simplified version of this would be, in the absence of amino acids, SZT2 is this purple scaffolding molecule, and it does not bind to mTOR, which leaves mTOR sort of just diffusely localized in the cytoplasm where it's inactive. But if you then treat cells with amino acids, mTOR is recruited to SZT2 at the lysosome where it is activated and functions. But in the absence of SZT2, even in the absence of amino acids, mTOR is always localized to the lysosome and is constitutively active. And so we just did an independent replication of the same study that was done in these two Nature papers. So it turns out that in wild type HEK cells, if you amino acid starve, the cells respond by reducing their activity of mTOR. And then if you retreat with amino acids, the phosphorylation of S6K by mTOR is restored. But in cells that are complete knockouts, and we did 2 independent clones, even in the absence of amino acids, mTOR is constitutively active. So this was sort of our first evidence that we could create a cellular model and sort of adapt this approach to VUS resolution. So what we did, we turned to classical CRISPR-Cas9 gene editing and basically co-transfected with guide RNAs and SSODNs that would allow us to generate cells that would have the exact variants that were identified in our patient cohort, and then plate them in 96 wells at single cells, grow up clones and verify the genotypes by Sanger. And then we could test function in our amino acid starvation assay. In just showing one example of one of the variants that we tested here, this in-frame deletion, valine 1984 del, we can see similarly to the complete loss of function variants that in the absence of amino acids, the activity of mTOR is elevated even in the absence of amino acids. We also did this for an additional variant, which was a predicted splice variant, and very similarly saw constitutive mTOR activity. And so this was exciting to us at the time, but was also a little bit intimidating because this took months of effort of cloning and validating guide cutting and creating clonal cell lines. And the idea of doing this for all 12 of our variants identified in our cohort was a little intimidating, plus the fact that eventually we would want to scale up to do additional variants. So we set out to find a way to at least increase the throughput, maybe not to the level of a MAVE that we're still working on, but something intermediate between this and a MAVE. So this is what we came up with. We started again with diploid HEK cells. So just like before, we transfected them with a single guide and SSODN for editing cells to have a patient variant. And what this left us with is, even without cloning into a 96 well plate, we had a pool of cells that had basically, by and large, these only 3 different combinations of SZT2 variants at a genotype level. So by far, the most common outcome was a cell that would have 2 loss of function variants, 1 on each chromosome. And this is because under our transfection and selection conditions, we had roughly 60+% chance of non-homologous end joining that would generate these variants. The next most common outcome were cells that were compound heterozygotes. So they would have 1 loss of function variant caused by NHEJ, but they would have 1 copy of a patient variant induced by homology-directed repair, HDR. And then very infrequently, our edit rates, just for anyone interested, were anywhere from 15 to 30-ish% on which varied a bit from transfection to transfection and from target site to target site. But yeah, rarely we would get cells that were homozygous for a patient variant. Then we would amino acid starve the cells, fix them, stain for phosphorylated S6 as a readout of mTOR activity, and we could perform fluorescence activated cell sorting to separate cells with low mTOR activity from cells with constitutive mTOR activity. And the way we envisioned this assay working is if the patient variant was a functional, encoded a functional protein, that would be sufficient to rescue amino acid sensitivity of mTOR, because this, again, is an autosomal recessive condition. One good copy is sufficient. So essentially, the purple cell in this case would be present more in the low population than the high population. Conversely, if the patient variant was encoded a loss of function variant, this would fail to rescue, and these cells would be enriched in the phospho-S6 high cell population. So as a proof of principle, we went back to our cells that were complete SZT2 knockout, and we compared our parental controls, which when you amino acid starve them, the vast majority of the cells are sort of left shifted or present in the phospho-S6 low population, whereas cells that are complete loss of function for SZT2 are primarily right shifted, which indicates constitutive mTOR activity. And then if we mix the two cell populations and then sort them, we could actually then isolate genomic DNA from the sorted cells, perform PCR amplicon sequencing, and develop a scoring system for our variants. And so we ended up naming this CMAS or CMOS, which stands for Constitutive mTOR Activity Score. It's a very simple metric. It's literally just the percentage of alleles for a given SZT2 allele found in the phospho-S6 high cell group divided by the percentage of alleles in unsorted cells. And our thought process here was that a high CMOS would be indicative of a loss of function variant, whereas a low CMOS would be suggestive of a benign variant. So this we were able to do in all of the VUSs in our expanded cohort. And what we found is that 3 of the 12-ish variants had CMOS scores that were very similar to all of the loss of function variants that we generated by non-homologous end joining. So these were our internal positive controls. Whereas the vast majority of the VUSs had lower CMOS scores that were closer and more similar to our high minor allele frequency nomad control benign variant. So this was encouraging to us that this assay can be useful at least to discriminate loss of function variants. I do want to give the caveat that some of these variants do have sort of intermediate CMOS scores that could be indicative of sort of a partial loss of function, but I don't think this assay as it currently exists is really sensitive enough to pick out partial loss of function away from complete loss of function. So we're still labeling these as more likely to be benign than loss of function, but I also would be cautionary against sort of trying to call these truly benign. We also don't know if partial loss of function variants can even cause a phenotype. That's an open question at this point. So going back to our cohort briefly, one thing I didn't highlight before is we actually had 3 individuals that had a recurrent variant, this Bailey 1984 Del. And we thought that was quite interesting. And going backwards, it was one of the few variants that actually had loss of function as the sort of the outcome from the functional experiments, both from the higher throughput version and the lower throughput version. So we sort of asked ourselves, like, I don't know, this was ringing bells to me, like Strata and PMSC, like, could this be a founder variant? Like, why is this one variant recurrent versus the fact that this is a gigantic gene and most of the variants are sort of evenly spread throughout the gene? So we went back to Nomad and took a little closer look, and I couldn't believe that I had missed this earlier. It turns out that in the Ashkenazi Jewish population, which isn't a widely sequenced population in Nomad, but decently sequenced, the minor allele frequency for this variant is roughly 10-fold higher than for any other population sequenced in Nomad. So this suggested the possibility of a founder effect to us. So then what we did is we went back and looked at the exome sequencing data that we had on the 3 individuals in our cohort that all shared this variant. And we were able to identify a shared haplotype that's roughly 4 megabases in size. I'd love to be able to narrow this down further, but there were just only so many SNPs that had reasonable minor allele frequencies to do this kind of approach. But by and large, somewhere in the 4 megabase size range that was shared between all 3 individuals, despite these individuals being, as far as we could tell, unrelated. So sort of this suggests to us that the carrier frequency in Nomad is actually, relatively high for this variant. And so this is a variant that we would think of as worth including on genetic screening that's specific to this population. So that's something we'll probably pursue is reach out to those testing companies and see if they're interested in including this variant. So the last thing that we did for SZT2 was take a look at sort of the, we did the hypothetical of what if we did reassign ACMG scores based on our functional assay, what would happen? And so at the beginning, before doing any work, we did have one individual who had pathogenic or likely pathogenic variants in both alleles, but after functional testing, 5 out of the 12 individuals would potentially be categorized as having pathogenic variants on both alleles. And so, this suggests that by doing this kind of approach, you can identify additional candidates for precision therapy approaches, such as there's currently mTOR pathway inhibitors that are under investigation, such as sirolimus or everolimus. And then conversely, all of the other individuals previously had at least one VUS, and now based on, if you were to adjust their classification based on the functional testing, all of them would have at least one likely benign variant. And so for all of these individuals, we would potentially recommend further genetic testing that SZT2 is unlikely the answer for what's causing their phenotype. So in summary, we developed a functional assay that can at least identify pathogenic variants in an mTORopathy gene, SZT2. We identified a recurrent variant that's likely a founder in the Ashkenazi Jewish ancestry population. Things that we'd like to do in the future, I'd love to be able to adopt this for a more neuronal cell type because a lot of the mTORopathies have neuronal phenotypes that we can't really assay in a HEK cell. HEK cells are great because they're easy to grow and they transfect really well and they're just generally easy to work with, but they do have limitations. Biggest of all of them is increasing throughput. This is not a MAVE and I would like it to be at some point. So that's sort of what I'm working towards at the moment. And then we think this has applications beyond SZT2. I think it's a sort of a general approach that could work for other genes that are mTORopathies. And again, the potential benefit of this is that you could identify individuals who could benefit, assuming mTOR inhibitors ever get approved for treatment of mTORopathies. I'm going to really quick switch gears. I know we're running out of time, but I did want to mention a little unpublished data. So we've recently started trying to apply this approach outside of SZT2 to see if it is viable to do this kind of work in other genes in the mTORopathy sort of spectrum. And so the first one that we've tried is in TSC, or Tuberous Sclerosis complex. I won't, just because we are running time, I'm going to skip the background, but just saying that TSC2 is an autosomal dominant disorder and otherwise shares a lot of overlap with SZT2. So what we decided to do in this case, because this is an autosomal dominant disorder, we switched to haploid cells instead of the diploid HEK cells. That would be kind of hard to interpret. Did a very similar transfection guides and SSODNs, just targeting at TSC2 instead of SZT2, and then tried to FACS sort to see if we could distinguish cells with loss of function compared to unedited cells. So this is work of a talented undergrad in the lab, Karina BR. And this is ongoing work, so this is going to be her honors thesis. So I don't have a ton to show, but I did want to mention that we were able to see that, at least by FACS, if you compare HAP1 control cells, these cells, even without amino acid starvation, mostly have relatively low mTOR activity. So you can see they're left shifted relative to this standard red line that I'm drawing here. Whereas cells that were transfected with a TSC2 guide RNA that we validated that we does indeed cut and disrupt the gene, these cells are primarily right shifted into sort of the PS6 high group. And then we do have an N of 1, so take it with the appropriate grain of salt, that the a known pathogenic variant, RH611 glin, does actually seem to be enriched in the PS6 high group relative to unsorted cells. So a lot more work to do here, but encouraging that sort of this general approach will work in genes outside of just SZT2. So with that, I will stop and thank my lab, who are all amazing to work with, our awesome clinical collaborators, both on the adult side and the ped side, our international collaborators who helped us gather the cohort that worked out, really helped us understand SZT2 better, all the patients who helped us as far as just making this research even possible, and then our funding. And lastly, I'll just put in a little plug that we're always recruiting. So if any of what I talked about today sounded interesting, please reach out and get in touch with either my mentor, Gemma Carvill, or myself. We're especially looking for postdocs and technicians to work on this kind of stuff. So don't hesitate to reach out. And thank you so much for everyone's attention.
Great, Jeff. Thank you for that excellent presentation. I actually really liked how you stepped us through the underlying genetics at the beginning of your talk. I think we should have time for a few questions. You can still add them to the Q&A, so we'll let that go for a bit. And I might just kick it off with a question. So you mentioned, right, that you're working on this MAVE using this phosphorylation readout, maybe scaling this up. And yeah, I may have missed this, and I'm not an expert on phosphorylation readouts, but I was curious about like the current bottleneck. You kind of hinted at this at the end, but I mean, it seems pretty possible.
I think so. I don't think it's quite as straightforward as a cell survival or a cell depletion kind of approach, which I would love to have, but just don't have that luxury for these genes. But I do think it's possible. I think the hardest part is I just learned how to do CRISPR-Cas9 like for this approach. So trying to learn how to do the libraries that we need to do saturation mutagenesis, for example, I haven't gotten there yet, but that's what we're working on. So yeah, I actually quite like Steve Irwood's saturation prime editing approach because we're mostly interested in missense variants more so than it's nice to have some premature truncation variants as sort of positive controls, but we can do that with saturation prime editing too. And so if we can really enrich our library for as much missense variants as possible, that's sort of where we're leaning. I mean, we'll see how things go. We have a couple alternative approaches that we can try, but yeah, that's what we're trying at the moment.
Cool. And I guess by fitness readout, you said it's a little bit more difficult. Is it just maybe a little noisier for the phosphorylation readout? Is that what you mean by that? Or
so the biggest problem is just getting enough cells back. So say I transfect 10 million and that's what I take to the flow sorter. I rarely get more than 200,000 cells back in an individual cell pool. So trying to scale that up to do thousands of variants and make sure that each variant gets representative by, I don't know this is what I'm still trying to learn, like how many cells you need per variant. Just seems a little tricky, but hopefully we'll get there. We're definitely planning on it.
It seems like you're well on your way. And so I guess there aren't questions right now, but we're running out of time, so maybe I'll just wrap up. I just want to say a big thank you to both of our excellent speakers today and to all of you for attending. And just a final plug for our September seminar that we already have the speakers scheduled, and we should send out a flyer, I think, soon. So you should put that on your calendar. And thanks again, everyone.